Hi, good afternoon everyone, and welcome to the Botany Bistro. We're going to shake things up a little bit today and have a guest and also show some images today. So we haven't done that before, so I'm really glad that you're here to join us for that experience. While we have been doing this for 7 sessions, some of you might be new, and so I'm really excited to introduce the program to you. This is a free program that we offer every other Friday. Starts around noon, so you can listen to us on your lunch break and you can learn a little bit about botany. We tie botany into our everyday lives and learn a little bit about some of the foods that we enjoy and some of the plants that we like to see in our gardens. So the program that we're running is actually based in Cincinnati, Ohio. We are sitting inside the Civic Garden Center. We are a nonprofit that's been around since 1942. We were started during the Victory Garden movement, and we're very happy to continue to educate people who are passionate about nature and the outdoors. And we also love having volunteers. So if you ever want to come and volunteer with us, you can help us maintain some of our community gardens, work with students in schools. And right now we're really focusing on building up an urban restoration conservation program, thinking about native plants, soil health, and water conservation. So those things are on the horizon. This is going to be our second part of our plant anatomy talk. So last week we talked about plant anatomy and microscopy and how we can look inside these plants and learn so much more about them. And so I am very excited about my special guest, Dr. Dan Gladish, who I'll introduce in just a couple minutes to help us with part 2 of our plant anatomy. Your homework for last week was to talk about your favorite organelle in the cell, and so hopefully some of you posted that. But if you haven't, you can definitely go back to the archive and watch number 6, our session 6 on plant anatomy, and learn a little bit about what goes on inside the plant cells. So what's for lunch today? I have some delicious roasted potatoes here today.
Smells great, yeah.
I've got enough for both of us, so we'll be good.
Oh, fantastic.
And I wanted to bring a roasted root salad with me, but my carrots and parsnips weren't quite up to the task, so I grabbed some potatoes, and my friend Dan reminded me that
They are not roots.
they're not roots.
Yeah, they're stems.
And so that's just
Modified.
one of the fun, like, little botany tidbits that you'll get from watching the Bistro. And you can definitely show off your botanical knowledge and say, oh, are you enjoying those stems?
Yeah, that's what we call tubers.
Yeah. Now, sweet potatoes, on the other hand, are roots.
Yeah.
But this is one that kind of tricks people up a little bit. So I hope you're all enjoying lunch and having some good conversations with people about plants in your lives. And we're going to go ahead and jump right in. So I'm going to introduce Dr. Gladish. How do you want me to call you?
You can call me Dan.
I can call you Dan? Okay.
Yeah, we're old colleagues now.
We've known each other for quite a long time. Dan was born and raised in Southern California, graduated from school there, and then moved to North Hollywood. He was not always a botanist, and so I am always interested in how people get to their careers. And the more I ask, the more I find that it's not always a straight line. So Dan was an entertainer, very talented musician, and left the music business and worked on farm, was a carpenter, drove a semi-truck, and then went to undergrad at the University of California at Davis, where he earned his Bachelor of Science in Botany.
I want to emphasize that I was in my 30s by then. Yeah, so it's never too late to start.
Yeah, definitely. And I think that we've sparked some ideas in some people here on the program before. And so if you want to get into this, it's open for you.
Yes.
Yeah. He spent 29 years teaching full-time for Miami University. And that's where we met, on the Miami University Hamilton campus. And we were working together on a program for 3rd and 4th graders who were experiencing plants up close at the conservatory there. So if you have not been to Miami Hamilton and seen that conservatory, I highly recommend it. And that was a really cool project that we worked on together. Lots of impact for those students.
8,500 kids.
Boom.
Over the period of time till the school district yanked the transportation.
Oh, that's always such a challenge. We struggle with that too.
It's expensive for them, I guess.
Yeah. And so we're going to learn a little bit about his dissertation research on temperature and root development. And we're going to learn a little bit about some exciting new research. And I wanted to congratulate you on your recent retirement.
Last week?
Not that you'll be any less busy because you're going to continue your research.
Yep.
But I am very happy that you get to explore that in a different way. All right, so that's our guest. And if you ever want to get in touch with Dan, just let me know. You can communicate with me via our website, and I can pass on any additional questions. But remember that at the end of our talk today, I will open up that chat so you can save your questions, and then we can answer them at the end of the program. All right, so I want to let you know just before we dig in that we will be having our plant systematics and taxonomy session on September 1st. That'll be our next topic that we dive into. And we're going to learn a little bit more about binomial nomenclature and plant families during that session. And then your homework for today is going to be to tell us about your favorite root. And please share that on our social media pages. I think that would be fun. But those of you that are listening know that potatoes are not going to be on that list. At least Idaho potatoes. All right. So I've got a few questions for Dan, and then he's got some presentations that we'll share on our screen. But the first thing I wanted to ask was just what type of science/research do you do?
Well, I guess I've usually described it as developmental anatomy and physiology. Essentially roots. Root development and root anatomical structure development, how roots grow. Awesome. And what influences them. It's kind of sometimes referred to as ecological physiology. So it has different names.
Neat. And what drew you to using microscopy in your work? Because I've seen a lot of your work has amazing microscopy images.
When I was an undergrad, I took a class of plant anatomy, and I absolutely flat loved it. And it was all microscopes all the time. And the professor was wonderful, and the teaching assistant was wonderful. And I ended up begging the professor to let me work in his lab as a research assistant so I could keep doing this kind of stuff. And so that was what got me started with microscopy. It was just cool to look at small things and up close and personal, so to speak. And so that's the answer.
It's like a world within a world.
It sure is. And I've only done a little bit of electron microscopy, but my students have done a lot, and my colleagues, and I'll show you some microscope, what we call micrographs of some of that kind of work. My main collaborators now live in Japan, and so I have 3 colleagues there. They are all retired.
That's great.
But we're all 4 of us still working.
Yeah.
Because it's addictive.
Yeah. Well, it's nice that you have that team. And how is your research related to plant anatomy?
Well, it'll be kind of obvious once I start showing you stuff. I mean, essentially everything the way we have probed the questions that have arisen, and I have to say, I didn't have an idea of what to study. It just fell on me. It's like, oh, that's pretty peculiar. And it's like the old thing, the most important question in all of biology is, what the hell is that? So to search and uncover the answers to the questions that had started occurring as we proceeded required slicing and dicing them, getting in there and taking a look at the anatomy. And our latest research is a reevaluation of some classical theories about how meristems work, and in particular, we're working with cereal grass roots.
All right, well, let's dive into it. I'm excited to show you some of the things that Dan brought today, and we're going to share our screen. We've got two different presentations for you.
One is the old stuff and one is the new stuff.
Yeah, so we'll start at the beginning here with how it all began.
That's a real micrograph and longitudinal section of the tip of a root of some kind. I can't tell what the species is from the just looking. But here's an exterior cartoon showing the root cap down here, and this part right here, this is the part of the root that my colleagues and I have been studying for years. Up here in the part of the root that's stopped. In fact, you can't have root hairs or lateral roots where the root is growing because they get scraped off.
Oh.
Because the root is pushing through gritty old soil. Now, over here is an anatomical view, and it's highly simplified, but there's what is sometimes referred to, it's labeled here as quiescent center, but it's the promeristem. That's the very earliest places where the cells start to divide, and right in the middle of this place in most roots, the cells don't divide. They just sit there. If something injures or disturbs the balance of things, then they'll start to divide. And the cap is sort of a soft helmet, because remember, this thing is like a hydraulic ram being shoved down through the soil, and it's gritty. So they're young cells. They don't have time to get tough or anything like that, so they're sacrificed. As the root pushes through the soil, they get scraped off. They also secrete this stuff called mucigel, which you see indicated in this kind of gray cloud around here, that creates an ecological environment around the root tip that favors useful bacteria and fungi that actually help the plant. Okay. My very first experiment that I did for my PhD, I actually did as an undergrad. Okay, and it was an accident. I mentioned that I wanted to work in a research lab, and the professor, Thomas Rost, assigned me to help one of his grad students, and he was working on trying to extract DNA from the tips of growing roots to isolate genes that were specific for that process. And I thought I'm real clever, I just took biochemistry, and I thought I could make that easier for him, because in those days we didn't have PCR, polymerase chain reaction, which is used to amplify small amounts of DNA. It's used a lot in forensics, in CSI stuff, But that had not been invented yet, so we had to grow lots and lots of roots, and I thought it'd make it easier if I could make the root tip bigger by manipulating it with temperature. And it was a miserable failure. And so I had a bunch of beakers that were, let me try this. This is this kind of situation that I was looking at. I was just, hopefully everybody can see these long roots that don't have any branches on them yet. And that was sort of the situation that I was looking at. And I grew roots like this in beakers up against the glass at different temperatures. And it was midterm time, so I left them on the bench to clean up later, and came back, and I saw this amazing thing. The two beakers on the left, this is grown at 15 degrees Celsius, which is about a little under 60 degrees. So this is like a cool early spring temperature of the soil. And this is later in the spring when the temperatures would be around 75 degrees Fahrenheit. And what's interesting is that the primary root, which is or taproot as it's sometimes called, in peas, these are garden peas, Alaska is the variety, and they grew twice as long before they stopped growing at this temperature than this temperature. And these roots over here, this is about 95 degrees probably. in Fahrenheit, it's 32 Celsius. They've already stopped. They've stopped growing. They're only about 10 centimeters long. Okay, but the thing that was interesting to us was that the spacing of the lateral roots was closer at the colder temperature than at the warmer temperature, and almost no laterals formed at the highest temperature, which is why you can't grow peas in the summertime. They just don't develop a root system. So, and I like to play around. So here what we did, what I did was these were grown at the middle temperature, about 25 C. That's room temperature essentially. Okay. And I would put them in the warm temperature for a day, and they wouldn't make roots for as long as they were in that warm temperature, lateral roots. And these over here, these were grown like that for 3 days. So, and they would slow down because they don't grow very fast at 32 degrees. That is the primary roots. So anyway, this was an experiment that happened because I didn't clean up. And I went ahead and became the first couple of chapters in my first published paper that I wrote on as a first author. And then I started doing experiments involving flooding, because we figured out that it wasn't so much temperature as oxygen that was the problem. At higher temperatures, plants use more oxygen. And these pea plants have evolved to grow in cool temperatures. And so if you warm them up and then give them too much water, then they struggle. And there's more to this story than just this. But this is grown under what we call field conditions, although it's not a field, it's a beaker with paper, and I got some vermiculite medium in the inside, and the roots are growing next to the glass between kind of a sandwich. These plants over here were, I stuck the seeds in there, and then I just flooded the beaker. And you can see those roots do not like to grow down into that stagnant water. But they will make a lot of lateral roots.
Interesting.
Under those circumstances. Okay, so now, if you grow them under the normal conditions, like in the first beaker I showed you just a second ago, this is how much the roots would grow in 1 day. And that's about 2, maybe 2.5 centimeters.
That's amazing.
It's pretty fast. But if you flood them suddenly, they're growing along happily, and they're relatively short to start with, they slow down, but they keep on growing. That's 2 days worth of growth, about 1 centimeter a day. But if you let them get long and then flood them, they do this really weird thing. Hopefully you can see it. The tips often curl up. They make like a pig's tail, or they just stop growing. Why? Well, it turns out that, let me show you. This is a picture of an anatomy of a normal cross section of a pea root, kind of in the middle of the root. So it's normal anatomy. This is the xylem sector right here. These are the phloem sectors where the food is coming down. This is where the water is going up. And this group of cells in the middle eventually develop into what are called late maturing metaxylem vessels. So they're big and they have a large capacity to carry water. However, if they're oxygen stressed a little bit, these cells in the middle kill themselves. Now I noticed I said kill themselves. Well, we didn't know that at first. They would just make a hole like you see over here. And I got another slide here. So this is two micrographs showing a longitudinal section in the middle of a pea root. And this is shortly after flooding. And this is a little later. This is maybe 6 or 8 hours after flooding. These things open up really, really fast. They're not dying. If they died, the cell walls would all still be there. And so we started suspecting that this was a programmed response. Something evolved to help these little seedlings. If they get flooded in the spring and it's a little bit warm, they can make this hole. And here's another picture. Sometimes they're really quite big. And it turned out we learned fairly quickly after that that was not full of water, that was full of air. Okay, so here's the normal xylem sectors right there. There's a hole in the middle, and this hole actually formed over on the side. So we called these vascular cavities. And here's a scanning electron micrograph that was made by my graduate student. Her name is Prabhasha Sarkar. And you can see that there's nothing in here at all. So the cells didn't just die, they destroyed themselves. And Prabhasha was a good electron microscopist, and she took these TEMs, called transmission electron micrographs. These are normal cells right here, where 3 cells have come together. That's a... Okay, I think we're okay.
Yeah, I'll just do this thing. There we go.
Okay. So that's a junction between these 3 cells. This is the cytoplasm here. It's really thin in the other 2 cells. But these walls are being destroyed. So it turns out that in programmed cell death, in some cases, well, in almost all the cases we know of, the DNA changes its program, produces a bunch of enzymes, and then one of the products of those changes is a DNA enzyme that destroys the DNA. The nucleus falls apart. The vacuole, did you tell them about vacuoles?
We did.
Yeah, the vacuoles rupture and the cell dies. Okay, in this case, that destruction process included enzymes that made the walls disappear. So that's really pretty cool. We studied the effects of flooding in programmed cell death, and Prabhasha got her PhD doing that stuff, figuring out all the biochemistry and so on.
I love that it was a mistake. It started out as a mistake.
Yeah,
and
so
then...
serendipity is one of the great tools of science. The most famous story is Fleming and the discovery of penicillin. He didn't clean up on Friday and came back, and all the bacteria that in his plate had been destroyed by this fungus that had contaminated the plate. So anyway, it's a great story.
That's so neat. And I use peas often in my programs because they're easy for children to handle and they have a quick germination rate. But definitely I'm not going to be grabbing for them in the warmer months. You know, that's when you're going for your beans.
Yeah, use beans.
Yep.
Kidney beans.
It's almost time of year to put them back out, and we've had quite warm temperatures. I mean, we're definitely dealing with a heat wave right now, so I think this will be really telling for us to start taking temperature of the soil before we're putting those fall crops in, because it might delay our start to that fall garden.
Yeah. I've been trying to grow lettuce in little planters.
Yeah.
And they don't like it at this temperature.
No.
They will start, but then they get diseased and stuff because they're not adapted to the temperature. Okay.
So that was the start of some really exciting research, and Dan didn't stop there, and so he's going to be showing us some new, very current work.
Yeah, some of the latter images that I showed you are from papers in which I collaborated with a Japanese colleague. His name is Teru Niki, and he was a professor at Takashoku Daigaku. That's Takashoku University. And he's now retired, just as I am. But so I wanted to mention him, and we teamed up with a retired computing engineer. And so we've undertaken now a whole new program of study using computers in combination with anatomy. So that's what the next thing is I'm going to show you as soon as Mary Lee fires up the...
Let's go for it. And this was a big catalyst for having Dan on the program. We randomly bumped into each other at an event, very surprisingly, but you know how the universe brings people together. And he showed me a little bit of this research we're about to see, and I was blown away. So this is very cool, very new stuff. There might be some concepts or terminology that are a little bit complex that you might need to do some research on. But I just say, let's go for it and we'll soak it all up. So,
Well, I'll try to demystify it as much as possible, and you can ask questions.
that sounds great.
Okay. Okay, now this is a longitudinal section view, and it looks like a cartoon, but it's actually made from an actual microscope slide. And it's cut the long way. And right down here, well, this is the vascular cylinder we were just talking about. And this line right here, that's the pericycle, the outer layer of the vascular cylinder. And here's the outside of the vascular cylinder, and there's the tissue that'll become the epidermis of the root. And down here at the bottom is the root cap that we talked about earlier. So I'm just reorienting everybody, and that's where all this research is focused on. We are reevaluating the classical theories about how this part of a root meristem works, the apical meristem. Okay. Well, there we go. Okay, now this is a cross section, and this is the vascular cylinder right about here. And what I wanted you to see from this is the only treatment that this slide has experienced is a particular kind of stain. That's a generalized stain that we use to heighten the contrast of things. But you can hardly tell things apart. Down there at the tip of the root, the cells are so dense and so full of cytoplasmic stuff. There's no vacuole. It's all cytoplasm. And if you use this stain, it's hard to tell things apart. This is one of those big vessels. And in these grass plants, they start their formation right at the tip. Okay, and this was discovered before. This is not new. But if you use this enzyme called ribonuclease, which is like the enzyme that the programmed cell death cells use to shred their DNA, this shreds RNA. Now, so this slide was treated, and this was the slide that was next after this one. So these are paired. And we treated with RNase and then used the same stain, and you can see it makes it a lot easier to see the cells. And the nuclei were not affected, just the RNA. How much RNA do you think there is in a cell? It's unbelievable. Okay, so that's our approach to get started. Now I'm going to switch to longitudinal sections now. This slide up here is a slide just like the second one. It was treated with RNase and it's stained with the toluidine blue. And then my engineer friend got to work. He's an image engineer, and he started to simplify everything to kind of so that we're emphasizing just the cell walls essentially. But we used the original slides to figure out this is a vessel, a metaxylem vessel, and it will take many days for it to finish its development. And he's colorized the metaxylem vessels. We call them X or LMX. And there's the pericycle. And here's a layer of cells right at the tip called the plerome. And the cells just underneath those, well, it's not really underneath, it's above those. And we could say it basipital to this plerome layer. Are initials that are dividing over and over again to produce all the cells up here. Okay, so that's the idea, and we make a complete set of them. I know you can't see the details in here, but there are 60 slides here. They've all been essentially processed that way and colorized, and we use that to make a virtual cuboid. And that's what this is up here. Now this one was actually made with cross sections. You can use either longitudinal sections or cross sections. But and you can make the cells that aren't colored disappear. You can do it for a part of the cuboid or all the cuboid. You can then use the cuboid and actually section it with the computer. So this is an This was made from real plant tissue, but but what you got is inside a computer, and you can use that to regenerate. Now this is through the same cells here as we go down through the cuboid, but the section is from above. It's a longitudinal section, and that is an image of what the cells look like through that cut right there in something that doesn't really, or well, it did once upon a time exist, but what we have are cross sections, and we made this longitudinal section from a cross section.
So great, 2D to 3D.
Yeah, or actually 3D to 2D in this case, and then and back.
And back.
Yeah. Okay. So here again, you can take a look. You can make a slice through the cuboid in different planes of section and get different views, longitudinal views of what was the original structure of that root before we sacrificed it for this project. Okay, and then if you want to, you can kind of make it three-dimensional. You can eliminate all the cells you're not interested in, just the ones that are colored. And so if you look in direction B, that's what the thing would look like if you could actually see it in real life. Okay, and so there are different perspectives. And right now we're using this technique, and by the way, that work has all been published now, to study these important plants. This is corn. This is its ancestor, teosinte. This is barley, and this is rice. And we're essentially finding out in very fine detail how these cells divided and grew. And I think we're going to be able to make this work. Okay, here we go.
Are you ready?
Yeah, I'm ready. So this is one of those cuboids with all the cells we're not interested in essentially made to disappear. And you can look in 3 dimensions at all these 3 big, actually it's 4 big vessels. And if I can stop it from turning, there you can see the actual original slides can still be recognized in this 3-dimensional virtual image. And that's it. That's what we're doing now. We're playing with computers and microscopy and anatomy, and I'm having fun doing it.
That's so incredible.
I just don't get paid for it anymore.
So I have a couple of my own questions, and I'm going to open it up for you to ask questions as well. But really, I think these 3-dimensional models and the ability to kind of do X-ray vision into a root is something that's going to inspire the next generation of plant scientists.
I hope so.
My son is 6, and who knows what he's going to grow up to be, but he's very into tech, and he's very into manipulating things, and I think this is something that could really spark a passion in him to apply that love of tech to something that is very useful, especially with the crops you've chosen to focus on, agriculturally very significant.
Very important, yeah.
So I'm going to open up the chat.